Our first speaker today is Dr. George Sutphin. Dr. Sutphin received his undergraduate degree and master's degree from the University of Washington in aeronautics and astronautics, and then discovered he enjoyed aging. He then received his PhD in molecular and cellular biology at the University of Washington and completed his postdoc at Jackson Laboratory. He joined the University of Arizona in January of 2018 as an assistant professor in the Department of Molecular and Cellular Biology. He is also part of Bio5. His primary research focuses on molecular genetics of aging using C. elegans and mice as model organisms. His research aims to prolong lifespan and health span and apply the extension to diseases of higher risk with age, like Alzheimer's disease. Today he will be talking about the direction and progress of his research. So please welcome Dr. George Sutphin.
Thank you, Erica, and thanks for having me out today. So I want to start, as Erica said, I study the genetics of aging. I want to start by giving you a kind of brief overview of my path to science and to aging science in particular, and then I'll talk about kind of the broad overview of how our lab approaches studying the genetics of aging and take you through kind of one storyline that's come out of that process. Yeah, that might not work. Let's try this again. Now we're going. All right. What is this?
Thank you.
All right. So my path to science was really not really a path. I've always been interested in science. So it's something about if you're going to make a contribution, contributing to kind of advancing our pool of knowledge is something that will potentially have a big impact now to kind of all of humanity, but also it's something that you can do that will make a lasting impact. So I was always kind of interested in research and making kind of advancing knowledge. And early on, my dad, who was a bus driver, who was not a scientist, he was always interested in pushing education in science. And early on, he said, you should study genetics. That's like the way of the future. And so, of course, as most kids do, I completely ignored him. And in middle school, I kind of got interested in computer science, building computers, doing programming, that sort of thing. As I went into high school, that kind of shifted. I got in with a group of friends. I was a Trekkie, kind of a nerd, and got into space science. So through high school, I was interested in that, and during my undergraduate, I started, as Erica mentioned, I joined an aeronautics and astronautics program. So I studied engineering. I was involved in undergraduate research from about the end of my freshman year. I worked on 2 projects during my undergraduate. One was called the RAM Accelerator. This is an alternative to your traditional rocket-based space propulsion. Instead of putting something in a rocket and carrying your engine and your fuel along with you, you basically build a long tube up a mountain and fire projectiles directly into space to hold your accelerations off the ground. This is great for putting things like water and other supplies into space. The acceleration, I would not recommend getting into one yourself. Most organisms would not survive. The other project is called Vortex Tube. This is a system with no moving parts that just uses kind of the physics of moving air to collect liquid oxygen. And so liquid oxygen is one of the major components of space or rocket fuel. And so you could, instead of carrying that all from the ground, you can take a plane up into the upper atmosphere, collect oxygen, and then use that to further propel yourself into space. You get big cost savings. This is still kind of at the conceptual stage, so I did some work on that. And then kind of in my early graduate career, I moved in the same department, but into a project focused on a fusion reactor concept, so looking at developing new fusion energy. And that's what I did up until then. And around that time, I got interested in the idea of lifespan extension and working on healthy aging, extending the healthy lifespan of humans. I had always kind of had an interest in this in the back of my head, but it was always something that I just didn't realize that it had become real science. Kind of, you always hear about the quest for the fountain of youth and things like that. But it turns out that the aging science had kind of come into its own, and people were really studying it. And as I started to read about that, I realized I was reading more about aging science than my engineering work, and something clicked. I decided that that's what I wanted to do, and ever since then I've been studying, to one degree or another, the genetics and the basic biology of aging. So kind of the point of all this is during your undergraduate years that you can get a lot of interesting work and develop a lot of skills, and that doesn't necessarily end up being what you do long term, and that's okay. I've used, while my undergraduate projects and my early graduate work have nothing to do with what I study now, I still have used the skills, the quantitative approach to thinking, the engineering approach to thinking, and apply that to my science today. So why study aging? You see a lot of this in the news. So you want to live forever. There's probably a new article like this about every other week these days. There is a lot of investment in science. And kind of the pat answer to this question is that, oh, I want to live forever, right? Woody Allen's one of my favorite quotes. I don't want to achieve immortality through my work. I want to achieve immortality through not dying. So that's kind of the first order answer, right? The search for kind of the fountain of youth or immortality in our literature goes back to essentially as far back as we have examples of written language. One of the oldest pieces of writing is the Gilgamesh Epics, and he had a search for immortality in his work. So this is kind of a long-standing quest for humanity. On the other hand, today, there are a lot of general health and economic reasons for studying aging as well. If you look at our population structure, so this is how many people are in each age group as a fraction of the population. Then this is just looking at 1970 versus 2014, and this is worldwide data, but this trend is true for most kind of developed nations, including the United States, and more extreme in certain places like Japan. In 1970, most of the population was, under 30, a big chunk. It had this kind of nice pyramid shape with very few people reaching older ages. And since then, we've gotten really good at dealing with the causes of mortality in the early life. Things like accidents and disease, infectious disease, excuse me. We have better antibiotics. And so we've managed to push back a lot of the early life mortality. And as a result, our populations are on average older. So there's a lot more people in the older ages. And if you kind of look at another piece of data, if you look at a bunch of the major causes of death and major forms of disease that are currently killing people in the developed societies, you see that they have this similar trend. So we can have things like diabetes and heart disease and cancer. And all of these things don't occur much in young people, but as we get older, the incidence increases. And so if we combine these two ideas, what does that mean? Right now, if you look at the top 10 causes of death in the United States, this is, again, also true across many of the developed societies. I think 6 or 7 out of 10, age is the major risk factor for developing one of these diseases. The top 2, which make up something like 60 or 70% are heart disease and cancer, and of course these age and aging are the major driver of this disease. And this is reflected in the economic data. Just as we get older, the amount of money per person per year we spend on healthcare dramatically increases, almost fourfold from young adult up to someone who's over 85. And so there's both a major health cost that's increasing with our increasingly older population and an increase economically. Another way to look at this is one of my favorite webcomics, XKCD. Probability that what you study will kill you. And you see on the overside, most mathematicians don't die at their topic of study. Out in the middle, volcanologists or something like that. Yeah, sometimes a volcano will get you. But virtually everyone dies at old age, the vast majority of our population. So as someone who studies aging, I'm pretty sure that that's the likely cohort of what's going to get you. Okay, so one of the underlying ideas in studying aging is that we have all these diseases that have a major driver as how old you are. So if we can understand the basic biology and molecular and genetics and molecular biology that underlies the aging process, target those systems, we can potentially have a major effect on a wide swath of diseases simultaneously. This is kind of contrary to the normal approach in medicine, where you pick a disease and you try to understand that disease and develop a treatment that's specific to that disease. That's been very successful, but we're hoping that by studying these underlying processes, we can do even better and hit a large number of diseases simultaneously. And as such, kind of have a much broader impact on general health, lifespan, quality of life, etc. So my lab, as Erica mentioned, we primarily use the roundworm C. elegans and mice as model systems. It's great to study aging in humans, but there are certainly drawbacks, technical drawbacks to it. Humans live a very long time, so to study aging, it takes a while. And it's harder to control human experiments due to environmental variability and genetic variability. C. elegans, this roundworm lives about 3 weeks, so you can do full aging lifespan studies very rapidly. And we also have a lot of powerful genetic tools. We can knock out or knock down every gene in the C. elegans genome. And we have lots of tools for studying their biology. They're one of the best studied model organisms. Mice have kind of a similar story, and they're somewhat intermediate. So they're much closer to human biology than a worm. That's the drawback to worms. They're separated from us by about 1 billion years of evolution. So there's always a question, is what we discover in a worm going to be relevant for human biology? Mice are the way we get around that. So they share about 95% of our genome. They get a lot of the same diseases. The drawback relative to a worm is that they live about 3 years. So while a worm, you can do thousands of lifespan studies. In a mouse, you can't. It's much more expensive to work with, and they live longer, so you can't do quite as many. But they're short-lived enough that we can do some. So our approach is to start with human data. This partially alleviates the concern that something that you discover in a worm will not be relevant to a human. So we first start with the strategy of identifying genes that we think are related to aging in a human, maybe not causal. So for example, we look at genes that increase or decrease with age. We take those genes, use C. elegans to screen the equivalent genes in the worm, so we can knock down the gene to see if we can affect lifespan. At that point, we know you have a causal relationship between your gene and aging. We pick kind of the best hits out of that, try to figure out what the gene is doing at the cellular level, the molecular level. And once we have an idea of what it's doing, we take that into the mouse, confirm that our findings in the worm are working in a mammalian system, that we can also extend lifespan in the mouse, or improve health or targeting disease. And ultimately, we want to bring that back around and use these discoveries to identify new drugs or new treatments to extend healthy human lifespan. So I'm going to take you through one story today that kind of uses this approach. Just to highlight a couple of things, I mentioned my path to aging science. In the process, we developed a tool to identify the C. elegans genes that are equivalent to the human gene. That used part of my computer science interest and background. And we're just now getting online an automated system so that when you measure a worm lifespan, the traditional way of doing that is you take your 1,000 or so worms, and every couple of days you go in and tap each one of those worms on the head with a piece of platinum wire and see if it's alive. You do that until they're all not alive anymore. And that's very tedious, it takes a lot of time. So we're developing a robotic system that can do that for us automatically. And so this is taking my engineering background and I'm able to apply some of those skills as well. So just reiterating that point that however you get there, you can apply your skills in useful ways. Okay. So we started with, for this particular story, we started with a screen. All of these genes, we collaborated with a group doing human genetics research and identified about 1,500 genes that change expression with age across human lifespan. So again, these are genes, because they change with age, there's some reason to think they may be important in aging, but we don't know if they're causing any of the functional decline or health decline that we see with age. We took those genes. We took about For this particular study, we just kind of skimmed the top 100 or so with the biggest effect, or biggest change of age. And then we identified the C. elegans gene. So each bar represents one of these genes in the worm. And we measured we knocked down each gene and we measured lifespan. So you can see we've got a pretty good range of effects, knocking down some of those genes, decreased lifespan by about 20%. And at the top end, and actually most of these, I think 40 to 50%, so which is more than you get if you just randomly selected genes, increased lifespan to one degree or another. And the top few were in the 20 to 30% range. So these are the genes up on this end that we are able to knock down and get a positive effect on lifespan. So these are the ones that we now know have a causal role in aging, and we want to figure out what they're doing. One of these genes is called Kynurenine, and that's going to be the focus of today. So Kynurenine encodes an enzyme at the top, and it encodes an enzyme in this pathway called the Kynurenine pathway. And what this pathway does is it turns tryptophan, which is an amino acid. It's famous for making you sleepy when you eat turkey, because there's a lot of tryptophan in turkey. That's actually a myth. Usually it's the other 10,000 calories you eat alongside your turkey that probably make you sleepy. But tryptophan makes things like serotonin and melatonin, and so people kind of got the idea that if you eat a lot of tryptophan, you might generate these sleep-causing molecules, and that would make you sleepy. Probably not the case, actually, but that's where it's mostly known. So a bunch of tryptophan gets turned into protein, it gets turned into serotonin and melatonin and other things. Only about 10% of it goes to these kind of more commonly known processes. The rest goes to this pathway called the Kynurenine pathway, the end of which is NAD, which is a molecule involved in kind of energy metabolism. That's one of the ways we generate energy. About halfway through is this gene kinase called kynurenine, and it just turns one of the small molecule intermediates into another one. It actually does that at 2 different points in the pathway. And if you knock it down, you get about a 20 to 25% extension of lifespan. So this is just percent alive with increasing age. So you want this curve to go push to the right. And it turns out if you just look at what this pathway does, and look through the scientific literature, there's a bunch of hints in the literature where it's associated with a bunch of diseases of aging, neurodegeneration, kidney disease, cancer, and so on and so forth. So this gave us some hope that we're getting an extension in worms, but this is also of interest in mammalian and human aging. So the next step was to take a broader look at the pathway, see if it was just something about this gene or if there were more genes in the pathway extended by then. Turns out there's technical reasons we chose 3 genes to knock down in this pathway. All 3 of them gave us a similar level of extension, about 20% for TDO2, which is right at the top of the pathway, and about 30% for this other gene, which is kind of one step down the pathway, called HOW. Because we got a bigger extension for HOW than for Kynu, we decided to make this our primary focus of work and trying to figure out why it is that this gene is extending lifespan. One of the things we noticed while we were looking at this experiment, looking at this gene, is this is a normal animal with a normal copy of the HOW gene, and this is an animal who has that gene deleted. As the worms got older, we noticed they were turning red. So it was a little weird. One thing about worms is they're transparent, so you can see kind of all their internal organs. And it turns out that what HOW does is change this molecule 3HA into another molecule, ACMSA. Turns out if you just buy a bottle of 3HA, it's red. And so this kind of makes sense, right? You knock out the gene that normally breaks down 3HA, and they build that molecule up, and they turn red as they get older. So this creates a hypothesis. Maybe the buildup of 3HA is driving the change in lifespan. So we tested that. You can actually quantify the amount of red, and it's pretty dramatic because of, so that was just a demonstration of that. And we also, using a more precise and quantitative technique, mass spectrometry, you can see that none of the other small molecules in this pathway go up that 3HA does when you knock out HOW. It doesn't for those other 2 genes that we talked about, Kynurenine and TDO. All right, so what's 3HA doing? Well, if you just feed the worms, add 3HA to their plates, which I was showing right here, you can see it in the plates, and where the worms are living, and so they're exposed to it. As you increase the amount of 3HA, you get kind of a peak lifespan around 1 millimole. So just adding 3HA is sufficient to extend lifespan. That's evidence that 3HA is the relevant molecule in this case. And you get a very similar lifespan extension here if you just knock out HAO. So it seems like 3HA is the intermediate molecule for knocking down HAO in lifespan extension. If you start looking at what's known about 3HA in the literature, there was kind of a theme that kept coming up. 3HA prevents LDL oxidation. LDL is cholesterol metabolism, protected against atherosclerosis by preventing oxidative LDL uptake, protects against neuron death by reducing oxidative stress. So we kept seeing a reference to oxidative stress. So it seemed like where it's been looked at, 3HA has been good for preventing oxidative stress. So oxidative stress, our cells, as a byproduct of many processes, produce things like hydrogen peroxide or superoxide, which can damage many of the cellular components, protein, DNA, lipid fats. And so we call this oxidative damage, and this accumulates as we get older, and so it's thought that the accumulation of oxidative damage is a major cause of age-associated cellular functional decline and downstream the diseases that we get with aging. So if this molecule is preventing oxidative stress or limiting oxidative damage, that might be a way that it's improving lifespan, extending longevity. We tested that by feeding the worms this drug paraquat, which produces superoxide and causes oxidative stress. Either knocking down HAO or adding 3HA was able to increase the survival on that case. And it turned out that we were also trying to make hydrogen peroxide plates, because you can just add hydrogen peroxide, and that's another way to cause oxidative stress. And when we did that, the redness of the media went down. And so this was suggesting that maybe 3HA was directly interacting with hydrogen peroxide and causing the 3HA to break down and potentially the hydrogen peroxide to break down. And so we tested that explicitly, and it turns out that if you just add different concentrations of hydrogen peroxide to media on the x-axis, and then increase 3HA in the media, the hydrogen peroxide detected essentially goes away. And that's also true in live worms. You can detect how much hydrogen peroxide they produce, and with age that goes up. And we can knock that down by making 3HA or knocking out HAO. So it looks like 3HA is directly getting rid of hydrogen peroxide. So this is where we're at now. We have a model where HAO inhibits the 3HA by breaking it down, and 3HA is good because it can prevent oxidative stress. We have some other data that it can promote proteostasis, which is protein misfolding and damage. It can prevent that from occurring. And we think these processes are driving aging. Oops, I'm missing out. I missed my first one. We're also looking at where in the worm this builds up and how it builds up with age, because where this molecule is important for which tissue systems it might affect and when we get into the mouse work. We're just starting to do the mouse work by either giving the mice 3HA as they get older or knocking out these genes in the mouse. We have both these mice on the shelf now. It's going to be a couple of years before we have anything to tell you about that. And we're also starting to study different diseases. We're particularly interested in a couple diseases: stroke, Alzheimer's disease, and sepsis, which is a severe inflammatory disease. So we have a couple of ideas about where this we may be able to get specific improvement in specific disease models. So that's the story of how we got to Kynurenine pathway and what we're doing with it. We've also got a bunch of other projects and candidate genes that we're starting to study. So that's kind of what the lab does and what we do. Just like to thank everyone, current and former members of the lab, our collaborators, and of course all of the University of Arizona and other funding agencies that we have. So thank you. Happy to take some questions.
You're studying lifespan in the C. elegans. Is there any way to study health span in the C. elegans model as well?
Yeah, we do that also. That's a good point. So there's kind of this idea that you might be able to do things to make us live longer, but not any healthier. And so you kind of spend most of your added lifespan in the unhealthy, kind of frail state at the end of life. And so we can study that in worms. They kind of get slower and stop crawling as quickly as they get older, so you can measure how quickly they crawl. You can put them in a drop of liquid and see how they swim. There's a bunch of functional measures. And basically, for the most part, things that extend lifespan also extend the health span of worms. So that's good. It seems that it actually kind of across the board, most things that we see improve general aging, also improve lifespan, also improve health span. So I think that's a good point. And that it seems to not be as big of an issue as it potentially could have been. Yeah.
Do you know how the ratio of like the ratio between the lifespan of a worm and a human? So like theoretically, if you made a worm live 30% longer, if you could apply that same treatment to a human, would that translate to 30% longer lifespan for a human too?
The answer is we don't know. There are cases where interventions discovered in worms or yeast or other kind of invertebrate systems have been translated into mice. In some cases, you get about the same level of extension. Caloric restriction, for example, you can just reduce the number of calories that a person or a worm eats, and you can do about as well on a mouse as a worm. Other things, it seems like you can't do quite as well as a mouse. So I think that's going to be very dependent on what your intervention is and how well the specific process translates into a mouse system.
So why does the worm have a mechanism to break down the 3-HA? Like evolutionarily, it's there, although it's just that 3-HA is better if it actually just remains there.
Right. So that's a good question. That's kind of true of anything that might extend lifespan that we find, right?
Right.
We don't know. So this pathway is the only way that worms can make this molecule at the end of the pathway called NAD from scratch. They can eat other things that have NAD-like molecules that they can then make NAD from, but this is kind of the only way to make it out of de novo or out of whole cloth, as it were. So it's possible that this, if you get rid of this and then you put the worm out into the wild where it's getting variable food and variable stressors, that it can't survive if it can't make NAD. So that's one possibility. But the real answer is we really don't know. There are possibilities where in the lab they're in a very comfortable environment. And so in the natural environment where they evolved, that might not have been okay.
Have you compared the supplementing with the 3-HA and other well-known antioxidants like vitamin C or something, and look at the differences?
Yeah, so this is kind of a complication in any antioxidant model. The oxidative theory of aging, as it were, has been around it's like one of the earlier theories of aging, and to one extent or another, it has not proven to be as robust as people would hope. So you can overexpress or feed animals antioxidants, and sometimes it works and sometimes it doesn't. So if the primary mechanism here is antioxidant, we need to figure out why it's different from other ones. Some examples of what might be different. If you just overexpress catalase, which is another molecule that breaks down hydrogen peroxide, whole body and let it go, you don't really get much of an effect. But if you target that catalase to the mitochondria, which is the organelle in the cells that kind of generate the most oxidative stress, then you can get lifespan extension. So it may be that this molecule is in the right place in cells to have a big effect. Again, we don't know yet. Yeah.
You know, proteins are knocking out any other cellular data for any other.
Some of them do. So at the very end of the pathway, there's this gene called QPER right here. So in the worm, most of these genes seem to be fairly specific to this pathway, but in the worm, the gene in mammals, QPER carries out this reaction. In worms, it's a different gene that carries out kind of a major lipid synthesis function as well. And so in that case, we're trying to decouple those two things and figure out what effects are coming from the Kynurenine pathway part of the intervention and which effects are coming from the other pathway. So that can be a complication. For the most part, it doesn't seem to be in this pathway, but in other pathways it is. Any last ones? All right, thank you.
Thank you.
So today I have the pleasure of introducing my own PI, Dr. Stephen Cowen. And first he got his bachelor's degree from the University of Wisconsin, then came here and got his
PhD in psychology and minor in neuroscience. After that, he went to the Neuroscience Institute in San Diego, where he did his postdoc, and then came back again because he can't escape Tucson. And now he's a tenured assistant professor for the psychology department. In addition to teaching classes here, he also, of course, does research, and that is focused on understanding the neural mechanisms for, excuse me, sorry, for learning, decision-making, and navigation. And by understanding these mechanisms, he hopes to see how they disrupt the processes from Parkinson's, aging, and chronic pain. So please give a warm welcome for Dr. Stephen Cowen.
Thank you, Allison. All right. Allison will be a highlight of this talk too. She's helped so much. I know you're already embarrassed, but yes. I'm going to give a little overview. Allison gave you a little intro of my background. So I got to the University of Arizona and to neuroscience by way of Wisconsin. So yes, I did my undergraduate at the University of Wisconsin in Madison, but my degree was in business, in marketing and management, with a specialization in international business, which was kind of odd for a scientist. And so realized that I needed some space, that maybe I needed to rethink where I was going. So I joined the Peace Corps, went to Honduras, lived on a small island off the north coast for about 2.5 years, and helped install solar panels, do micro-business consulting, but it gave me the space to kind of reconsider where I was, what my kind of passions were, what my interests were, what was I curious about. And it kind of brought me back to science. Science was, if I had been more rational and thought about what I did when I was in high school and before, I kind of would have realized that's where I would have gone, but it kind of gave me that space. And so then I came back to the US, and I took this, I had a great semester at the University of Wisconsin, Milwaukee. Took a beautiful group of classes from artificial intelligence to psychology, linguistics, philosophy of mind, but I took one neuroscience class, and I just really loved it. The strangeness of biology and science and neuroscience. How odd it is that we are composed of billions and billions of jiggling fat balls with strange proteins embedded in the membrane that are electrical and chemical. It's amazing that we work at all. But it's just so odd. So I was fascinated by that. I also have somewhat of a background in computer science. I guess we share something in common with real interest in technology and building stuff and robotics. And so I worked in a number of labs in Madison doing computer programming, helped with a linguistics lab, develop computer models for past tense learning at the time. This is a long time ago. And then also worked in a hippocampal modeling lab where we modeled epileptic seizure activity in the hippocampus. And so that finally gave me the cred to come back here, come to the U of A, to study neural recording, to study neural activity in the hippocampus with Dr. Bruce McNaughton. We were looking at how groups of neurons coordinate their activity during waking and sleep to help us consolidate and form memories. And so I was just very interested from many things in how groups of neurons work together to get things done, with those things being decision-making, learning, memory. And that carried over to my work at the Neurosciences Institute in San Diego, where I was first a postdoc and then got into a faculty position there, where I did a number of pieces of research on decision-making and cost-benefit decision-making, and how the neural activity in the frontal part of your brain contributes to that. And then I made it back here, and so very happy to be back in Arizona. Always loved Tucson, and yes, and that's where I am. And I'll give you here. Physically, we are here. This is the third floor of Life Sciences North. Here's our summer crew. Emily, I didn't have a picture of you. I forgot. I think I forgot to ask for it. But anyway, next one. I'll get you a picture for you. Emily's here. Francesca, and this is our summer group. Lindsay, Tony, who's just on his way to LA very soon, and then Allison. I'm glad you didn't wear your mask. Yeah. Allison's doing her surgery right there, and Karen. All right, so broadly we're interested in the brain. How does the brain work? Big question. And a myth is that the brain is very organized. We kind of see these pictures, you find a grandmother cell, oh, this area is involved in visual processing, this area is involved in memory. Wow, the brain's just like a nice bunch of bookshelves. But no, it's not. It's much more like that. It's much more like a Jackson Pollock painting, where you have there's some structure, kind of, sort of, in the anatomy. You see these broad swaths of connections between regions, but there's a lot of chaos underneath. And somehow, both the anatomy and the neural activity gets organized despite or because of some of the structure in the organization of the brain. And so we're very interested in that question. How is neural activity organized so that we can do things like think and remember and decide? And there are a couple things involved in that we are particularly focused on, is how oscillations, which themselves are neural organization, but how oscillations might help, and I'll talk about that a bit, how oscillations might help organize or coordinate neural activity, and how neurochemicals, like dopamine, might also help perform that function. And also how these mechanisms of coordination go wrong as we get older or as we get diseases like Parkinson's disease or conditions like chronic pain. And so we're interested in both how they do the good things that we need to get by in the day and how they go wrong in certain disorders. So what do I mean by an oscillation? So an oscillation is a coordinated and regular repeating pattern of neural activity that can be transmitted usually from one region of the brain to another or within a region of the brain. And it can be the product of a number of things. And a single oscillation, like if you saw an oscillation occurring 10 times a second, like 10 times a second in the brain, it might be produced by a number of different mechanisms. Some of it might be driven by some external activity or some sensory activity. Others might be internally generated by the intrinsic activity of the neurons or the neural networks. We're very interested in kind of dissecting and figuring out the generators of different brain oscillations. Because we believe, and they're also clearly implicated in memory, learning, decision-making, and memory formation, and consolidation. All right. The other thing that we're interested in related to coordination is this chemical neuromodulator called dopamine. Dopamine is a very interesting molecule. It's only like 22 atoms, very tiny. And there aren't that many dopamine-producing neurons in our brains. So we each have around 80 billion neurons. A very, very, very small fraction of those neurons are deep, deep, deep in the brain. So dopamine's been associated with many things. Typically, it's this, oh, reward, pizza, happy, that's a pleasure molecule. And there's some truth to that. It needs some more unpacking than that. But it's much more interesting than just a reward molecule. It's involved in many things, believe it or not, lactation, or at least stopping lactation. So when you need to stop lactation because let's say your baby's no longer breastfeeding, dopamine's the molecule that turns it off. And it's actually the dopamine neurons that are deep in the brain that are involved in that. So it's not just reward processing. It's also involved in vision. There are dopamine neurons actually in your eyeballs, in the retina. They're involved in improving your night vision, your contrast vision. It's involved in motor activity, hence its connection to Parkinson's disease. Our ability to initiate, to get out of your chair, to get something done, but not only initiating motor activity, but in also sustaining effortful motor activity. And also very important in other things like decision-making, evaluation, risk-taking, and such. But related to the effort activity and this motivation, that's something we were very interested in. I'm not going to talk about it much, but we're very interested in the lab. And like how dopamine can allow you to sustain activity, effortful activity, over a long period of time. Yeah, like those guys. I mean, I don't know how much dopamine is in a hamster, but there's got to be a lot going on in there to illustrate next research project. All right, dopamine. So dopamine levels go down particularly quickly in Parkinson's disease. There's a little area in all of our brains called the substantia nigra pars compacta, and it's one of about 5 or 6 different nuclei in the brain where these dopamine neurons reside. And that particular area is critical for our ability to use dopamine to control our movements and to initiate movements and sustain movements. Well, those neurons when those motor symptoms appear in Parkinson's disease, about 80% of those neurons are gone. They're dead. And the rest of them aren't doing so well either. And so we know that the loss of dopamine cells in the brain is a critical feature, not the only, but a critical feature of Parkinson's disease. There are other areas of the brain that have different subsets of dopamine neurons involved more in decision making and learning. And we're interested in both of those subsets of neurons. But it's not just that, oh, okay, well, I don't have Parkinson's disease, I have nothing to worry about. We all have a little vested interest in studying how dopamine works and how it declines because it goes down with aging as well. And so if we all live to maybe 150 or 140, if we do fix that, we've got to figure out how to keep those dopamine neurons alive as we get older. Because even in the healthy individuals, perhaps through oxidative stress and other complicated factors, those dopamine cells, even in healthy individuals, decline as we get older. All right. Now going back to oscillations, the loss of these dopamine cells and dopamine is also correlated very strongly with the appearance of bad oscillations. In one particular one, it's this beta band oscillation. People will talk about oscillations, they'll throw all sorts of Greek letters at you, beta, alpha, gamma, and it's like, what? Why are they doing that? Well, they're just different signatures for the different frequencies of the oscillation. This is a beta oscillation, which is around 18 to 20 times a second. And the x-axis is time, so this goes around almost a little over a half a second here. And you see these ripples here, 1, 2, 3, 4, 5, 6, 7, 8, around 8 or 9 in a course of a half a second. So that's an 18 hertz oscillation, 18 times a second, so that would be called a beta band oscillation. You see this appear in Parkinson's patients, and it's associated with their severity of their symptoms and their degree of immobility that they have, and the inability to act or initiate movements. All right, bringing that to maybe a global picture of what we're interested in the lab. I've mentioned that broadly we're very much interested in dopamine and neural activity. We're doing a number of projects related to the mechanisms that drive dopamine release. I won't again talk about that much today. But we're also very interested in sleep and memory, and particularly the neural oscillations and neural activity patterns involved in how memories are consolidated during sleep. And then I'll seemingly switch gears, but it relates to Parkinson's disease, talk about ketamine and the oscillations that ketamine can generate. And all of this is done in both healthy, normal animals, but also in the context of Parkinson's disease and aging. All right, sleep. Sleep. We should all do it. In fact, luckily we all do, because it's really critical for so many functions. Beyond the brain. I don't want to be super brain-centric. Sleep's important for a lot of things that go beyond our mental health. But it's very important for our ability to form memories, to be less stressed, to make good decisions, and such. And it's about generally keeping the homeostatic, the health, the balance of your brain intact, for your brain not getting too excitable and to go into seizures and things like that. And so you need sleep to help keep the brain in a more calm, let's say, state. We're particularly interested in a particular oscillation that occurs during a particular phase of sleep in the lab. There's like different phases of sleep. You've probably heard of REM sleep or quote, dream sleep. You might have heard of slow wave sleep. Well, there's a particular brain wave called the sleep spindle. It's a little slower than the beta oscillation. It's a 12 to 15 hertz oscillation that occurs, it only lasts for about a second or so, but it occurs during that slow, deep sleep. And it's very much connected with memory formation or memory consolidation. And for example, after a new learning experience, you get more of these sleep spindles after the new learning experience. And it's connected to new learning of episodic memories, like knowing that you're here in this room right now on this day, listening to this talk, memory of an episode, but also in learning a motor activity, like learning a new pattern, a new piano riff, or how to play guitar. So spindles seem to be very important for memory consolidation of various types. And also we're interested in these spindles because sleep disruption in general and spindle disruption in a number of other disorders are associated with pathology. So like sleep disruption is associated with depression, aging, and many, many other things. Spindles are also very interesting because the circuits, of the few things that we've figured out in the brain, we can be relatively confident that we've understood the core elements of the circuits that generate these brain oscillations during sleep. And it involves the thalamus, the sadly named relay center, although that's good enough for now, of the brain, a loop between the thalamus and the cortex back to the thalamus. This kind of feedback loop generates these spindle oscillations. And so we kind of have an idea of the mechanism and the circuitry involved in it. So we're very interested in spindles. We're interested in Parkinson's disease as well. What's the connection? Well, first, Parkinson's disease, as I mentioned before, is basically a signature of it, is sleep disruption. In fact, even before the very first motor symptoms become apparent in Parkinson's disease, you see REM behavior disorder and other sleep disorders occurring up to like 7 or 8 years before those motor symptoms occur. And so REM behavior disorders, when you start acting out your dreams, you might have flailing arms and things like that. So the partner that might be sleeping next to you might get whacked in the head. It's not very good. So that's a very early onset condition with Parkinson's disease. There's a particular form of Parkinson's disease that our lab is interested in. It's called the LERC 2019S, G2019S form of Parkinson's disease. And of the forms of Parkinson's disease, most are, quote, idiopathic, and that they don't have a clear genetic origin. But there are a few, around 10%, that have a clear genetic cause. And this is one of them. And so we're very interested in that one. We've been funded by the Michael J. Fox Foundation for 3 years now to explore this particular gene and how it alters sleep in mice. In mice that have had this gene modified, or this exact mutation that you see in human patients in these mice. And then we're looking at how the sleep is disrupted and how neural activity is changed. But you might say, well, what does that gene do? Easy question. Hard answer. Hard answer because it seems to have this particular LERC2 gene has its little fingers in everything. Going from mitochondrial function, and they can also get to cellular signaling, to autophagy, so basically cell death, other things, inflammation, oxidative issues. And it's not just involved in the brain, it's involved in this all over the body. And so it has many different functions. How it relates to sleep and sleep spindles comes from a particular set of studies that were done in knock-in mice, where this gene, this particular mutation, was embedded in knock-in animals, and then we measured them. We didn't do this. This is another group in Kelly. Measured the excitability of individual neurons. This was in slice, so this was a little slice of tissue. This wasn't a behaving, moving animal. But what they found is that the neurons, specifically the cortical and striatal neurons in these animals, were more excitable. They tended to fire more often. They tended to send signals or emit glutamate, is what these cells do, more often. And so it suggested that this particular mutation increases the excitability and energy, essentially, of these neurons. And that led us, amongst some other things, to the general hypothesis that because of this increased excitability between neurons in the thalamus and the cortex, that same loop that's involved in generating these sleep spindles, between this kind of circular loop that generates the spindles, may be enhanced. And that circuit, if that's the case, there isn't much work done on LERC2, but if this excitability of the circuits is enhanced, we predicted that features of the sleep spindle would be enhanced, like they would last longer, or they would be higher amplitude, or they'd be an increased frequency. So the general features of the spindle that would indicate that they're more powerful, they're more oomph, that the circuit is more hyperactivated, would indicate that they're increased. So we explored that in the lab. Actually, Allison did pretty much, almost in the latest phase, pretty much every surgery, right? Every surgery. So Allison implanted surface electrode arrays into our mice in both hemispheres. So these are similar to what we call electrocorticograms. It's kind of like a souped up, better version of EEG, where you actually rest the electrodes on the surface of the brain to get really clean signals of electrical activity of the brain. And then measured the brain activity from those electrodes. And here's an example of a very brief little sleep spindle that we measured from one of our little mice. The basic experimental protocol was to allow these animals to rest, allow them to explore for around an hour or so, half an hour, and then allow it to rest again afterwards. And during that period, we would measure the brain activity related to either just a very relatively boring experience they might have had, or a very active learning experience, which I'll talk about in a bit. Emily actually did, back there, she's analyzing data from another set of data from this very same group, where we let these animals sleep for 20 hours. We're looking at more circadian level changes in these particular animals. So that's another story. All right. And yeah, here are the features we're looking at, the duration of a spindle or the amplitude of a spindle. And so here's the first set of data we had. Each dot indicates the value from one particular mouse. This was a pretty heroic set of experience. We have a lot of data from these animals, so that's a lot of implants. And yeah, that's a lot of implants. Wow. Looking at it here. Wow. Okay, so yes, yeah, that's a good job. All right, so lots of work there. So we looked and analyzed a lot of features. We have the frequency, duration, and in fact, at least in the first two things that were kind of our hypotheses, that the frequency and the amplitude be enhanced, there was absolutely no evidence for that. In fact, it almost seemed to go to the opposite in frequency. There was a small, but admittedly small, effect on the duration. It lasted slightly longer, not even like a quarter of a second, longer in the LERT2 animals. So there were some changes in the features. This didn't really pull out a huge effect. So we looked more at what these spindles are theoretically about, which might be learning or memory consolidation. So I didn't mention before, but these animals also had to do this little gymnastics task where they put on what's called an accelerating rotor rod. So if you've ever done the log rolling championships, I'm sure a lot of you have, you get in a pool with a big log, and there's somebody else in the log, you've got to kind of knock the person off by spinning the log. Well, that's kind of what this is, except it's not competitive, I guess. They just compete against themselves. But they're put on this rolling log, and it gradually accelerates, and you count the amount of minutes or seconds, seconds, that it takes for them to flop off. And there are little cushions down here, so they're all right. And then you put them back on, and they get better over time. They stand longer and longer. So it's a motor learning task. They're learning to kind of balance themselves and keep up with the accelerating rotor rod. And the really interesting thing that we found is that what was cool is that the wild type, the animals without the mutation, as they basically increased their spindle amounts from the pre-rotor rod experience to the post-rotor rod experience, so the sleep right before they were on the rotor rod and the sleep right after the rotor rod, those animals had more spindles afterwards, which is what we would have originally predicted from any animal, is that if you're learning something, you're going to have more spindles after the learning experience to help you consolidate that memory. And this has been replicated and shown in humans down to rats and mice. But this is a novel task, so that was really neat to see that effect really strongly in the red, the wild type. There was a significant correlation in both groups. The better they got the rotor rod, the more spindles they had. So latency is how long it took them to fall off the rotor rod. So the longer, the better, the more they learned effectively. However, that effect wasn't present in the LERT2. Even though they had slightly more spindles overall, they weren't changing as a function of learning or as a function of experience. So there's something interesting going on there. So what are the upshots from all this? They don't seem to change much. Yeah, right. Okay, so the effect is that in wild type, in your regular like mice, spindles improve or increase with learning. But this isn't the case in the LERT2 mutation, even though they have slightly more. And this might be interesting, and Michael J. Fox is particularly interested, their foundation is particularly interested in finding new indicators on human subjects who have this mutation that tell them whether or not they're at the early stages or if they're going to express Parkinson's disease. And so this would indicate that if you're going to find any brain level indicators, like in spindles, that would tell them, well, this person's at risk maybe for Parkinson's disease and that they have this mutation, well, then you might need to challenge them with some sort of learning experience in order to identify or to create changes that are actually detectable from your measures. So, I mean, a human rotor rod? No, I don't think humans would agree you put on a rotor rod. But maybe something like it, some motor learning task where to teach them a novel, maybe finger tapping task or some sort of task and measure sleep before and after. And that might be the way to pull out differences between human patients with the LRRK2 mutation and not. So it kind of gives some ideas as to maybe how we design our behavior or how our experiments in humans. It might not be good enough to just measure their sleep. You might actually have to give them a learning experience ahead of time. All right, this one I'll keep a little more quickly, go a little more quickly. It relates a bit to our interest in Parkinson's disease, and that's our interest in ketamine. Now, ketamine is a drug that's been used, developed back in the late 50s as a general anesthetic. People know it as a horse tranquilizer. It's used commonly in the vet clinic for dogs, cats, and other animals, but it's also used in children because it's very safe, and it actually doesn't have some of the respiratory side effects that you might get from other general volatile anesthetics. And so ketamine has been around for a long time, pretty safe drug. It's also a drug of abuse. It's not terrible here, but in the UK and China, it's abused a fair amount. But ketamine is interesting for some other reasons, especially in the last 15 years. It's received a lot of press. Oh, 5 minutes. Okay, got it. It's received a lot of press because it's been linked to reducing depression. It's been used for the treatment of post-traumatic stress disorder, treating chronic pain, and in particular, we're interested in how it reduces dyskinesias, which are these kind of ballistic, flailing, uncontrolled movements associated with Parkinson's disease when the patients keep taking their medication. Because over time, over years, the medications themselves in Parkinson's disease cause problems pretty much as bad as Parkinson's disease itself. And so we are interested in how ketamine might serve to disrupt those ballistic movements. All right, these moving right along. So what we did here is we were looking at the neural circuits involved in Parkinson's disease and dyskinesia by implanting arrays of electrodes deep into the brain, and then measuring the brain oscillations like spindles, but these weren't spindles. We were looking at ketamine-induced oscillations. I'll go over that. I'll skip that for now. Okay. And so we measured activity over long periods of time, over a 10-hour period, in these different brain regions to see what ketamine actually did to neural oscillations in the brain. This is something that people don't really understand very well. And if you see here, here's motor cortex, this area about here involved in motor control. Here's the striatum, an area involved in both motor initiation and also decision-making and reward-driven learning. And here's the hippocampus, an area of the brain involved in learning and memory. And if you just eyeball this, you can kind of tell that ketamine has its own signature in each individual brain region. That ketamine does something very different to how neurons are coordinated in each brain structure. And this was surprising. It doesn't do the same thing everywhere. Frequency is on the y-axis, time is on the x-axis, and these are the oscillations that are induced by ketamine. You get this 50 hertz oscillation in motor cortex, super, super high frequency, 150 hertz oscillations in the striatum, and broadband, wideband, I would call neural noise in the hippocampus. So what it's doing to the brain depends on the brain region you look at. And this might be very interesting in that this narrowband synchrony might be very important to interfere with the patterns of oscillations involved in dyskinesia, which are near but not the same as these particular oscillation patterns to depression. And perhaps it disrupts these depressive, self-referential negative memories that keep cycling. It's very much a broad hypothesis. To disrupt some of the patterns involved in depression. All right. So what we're doing now, and so the last step, is we want to find out, oscillations are great, we love our oscillations, but what we'd love to know even more is what types of neurons or what neural circuits in the brain are involved in generating these oscillations. And so right now we're implanting more electrodes and just did a set of experiments. Allison actually did the surgeries for the last one, where we're going to measure the activities of individual neurons and inhibitory neurons and excitatory neurons to see what specific individual circuits are involved in generating different oscillations associated with ketamine. All right, yes, so we implant electrodes deep in the brain to measure single neuron activity. All right, so to summarize, we're very much interested in dopamine and neural activity, sleep and memory, and ketamine in the context of aging and Parkinson's disease. And all this work is done by kind of these heroic efforts, people like Allison and others, and Emily in the lab, and also from our collaborations with many others in our lab and also in other labs, the FOG lab, the Pareca lab, and of course support from our funding organizations, Ignite, Michael J. Fox, NIH, and the NSF. All right, thanks a lot.
Time for a question?
So if you've noticed that there's a decrease in dopamine when it comes to the onset of Parkinson's, is there also a link to like depression as kind of a pre-onset to a patient having Parkinson's? Or is it
That's a really good question. So pre-onset, so depression is definitely associated with Parkinson's disease. And reduced dopamine in the nucleus accumbens, which is another area. So Parkinson's disease is associated with dopamine loss, usually with circuits associated with motor activity, but there's another set of dopamine neurons projecting to the decision-making emotion areas called the nucleus accumbens that usually die later. And so one of the ideas, when you ask a good question, because not all Parkinson's patients are the same. Some may have death to different neurons at different time stages, but perhaps the usual course is that depression kicks in after late stage or at late stage Parkinson's disease, not before. But that's a good question. I wonder if there are just different patient populations that might actually show the difference depending on which neurons the subsets die first. Other questions? Okay, thanks so much.